and gentlemen, welcome to the evening forum today. Our issue for today is whole genome sequencing. First of all, let me introduce to you all our panelists for today. Right on my left is our specially invited Miss Jo, while on the right side is Miss Joyce.
Thank you for having me today.
I'm also very glad to be here today.
Without wasting time, I would like to seek information from our panelists for further details about the topic.
Okay, before everything, as we know, each organism has their own genetic code or genome that is made up of nucleotide bases A, T, C, and G. This is including bacterial, plants, or mammals. As a result, whole genome sequencing, also known as WGS, can be utilized as a thorough way to examine all genomes, and it became available after the publication of the Human Genome Project, which generated references for human genome sequences.
Okay, whole genome sequencing, or we can call as full genome sequencing or complete genome sequencing, actually is the process of determining the whole DNA sequence of an organism's genomes at a single time. So, whole genome sequencing has been used as a useful tool, such as an identify inheritance disasters, tracking disease outbreaks, or being used to conduct research.
Thank you for the information. Can Miss Jo or Miss Joyce provide us with a case study related to WGS to expand our understanding?
Well, based on a study, a 13-year-old boy was tested positive for renal tuberculosis disease. It was found that the child's father was diagnosed with TB when the child was only 1 year old. It was shocking that the development of active TB in a child after 12 years of exposure. Thus, in order to investigate the cause and the development of active TB, the use of the WGS provides resolution include diagnosis, treatment, surveillance, and source investigation of Mycobacterium tuberculosis infection.
Yes, I'm very glad with Miss Joyce. The use of WGS really provided the ultimate resolutions for the detections of tuberculosis transmission and reactivation events with the help of genome typing. The results show that the genetic distance between the father's sputum Mycobacterium tuberculosis isolate and the child's bronchial aspirate sample was not much difference. By applying WGS, we are very glad to confirm the epidemiological link between the two tuberculosis cases and point to the latencies of infections in the child.
That sounds incredible. Now, we move to the next question. Miss Jo, what is the status of WGS in Malaysia?
Well, the awareness of this importance of genomics has been lacking for the past decades in Malaysia. The application of genome sequencing technology in Malaysia is limited because the setup and maintenance is cost a lot. Besides that, Malaysia also significantly insufficient lack of the appropriate technical expertise in this field. Most of our genomics research in Malaysia are outsourced to other developed countries such as Singapore, China, and United Kingdom. So I cannot say that now WGS technique is well developed in Malaysia. It just needs time.
Thanks to Miss Jo, I would like to ask Miss Joyce, is the application of WGS study on renal tuberculosis still under research or under clinical trials?
Okay, for what I've known, the application of WGS or renal tuberculosis is still under research by a biotechnology research institute in University Malaysia Sabah. In their research, they sequenced the genome of Mycobacterium tuberculosis isolated from a patient diagnosed with TB. They used WGS to determine the source of survival, resistant of Mycobacterium tuberculosis with the help of a tool called Microbial Genome Annotation, also known as RAS.
I see. A very interesting sharing. Miss Jo and Miss Joyce, is there any possibility of applying WGS technology to local situation of Malaysia?
Yes, WGS could be the best-in-class genomic sequencing solution to investigate foodborne outbreaks. As in Malaysia, there were approximately 9,400 food poisoning cases reported in Malaysia in 2022. Whole genome sequencing could be a revolution for public health surveillance and help food manufacturers understand the origin of punctual or recurring food contamination by pathogens and to prevent potential future outbreaks.
Yes, for example, the outbreak of pandemic in Malaysia. Actually, Malaysia has been used WGS techniques to analyze the sequence of coronavirus, which provide insight into the virus from the Alpha to Omicron that circulating in Malaysia. Whole genome sequencing was performed for 204 SARS-CoV-2 from a COVID-19 cases and an additional 18,667 of SARS-CoV-2 genome sequence were retrieved from the EPICOS database for clade, lineage, and genetic variations analysis.
Thank you. As far as we discussed, is there any ethical issue or health issue of this technology?
In my opinion, one of the ethical issues raised by the WGS is about the informed consent. Informed consent can be understood in a very strict ways. The individuals are informed so they can make a well-considered decisions on consenting to a proposed intervention. While in absence of their explicit permission, no actions are undertaken. However, no mention of secondary use of genome sequence was often made. Can you think, can a person consent to something they have no details about? Okay, these tensions between the desire to support research in the public and the needs to protect individuals' rights and establish ethical norms has become a major issue.
Whereas for me, despite excitement about how WGS might revolutionize disease detection and prevention, there is concurrent concern that its introduction into clinical care, particularly of healthy individuals, it might cause patient anxiety and harm and increase healthcare costs. It was found that about 1 in 5 healthy adult patients who were sequenced have an unrecognized variant with potential risk for a Mendelian disease.
Thank you for the informative and interesting talk. Finally, we come to the end part of our session. We would like to say thanks again to our panelists for the interesting sharing. Hopefully this session will be beneficial for everybody. Thank you for your attention.
Thank you.
you.